Welcome back. Oh, it worked. So I'm sure I don't need to convince you all that mean and peak temperature have increased and will continue to do so in the upcoming years, and the consequences thereof include expected biodiversity loss. And recently there has been this body of work showing that in arthropods, high temperatures lead to lower reproductive performance. And essentially, the take-home message of all these studies is that heat-induced sterility should be a research priority. And I'm showing here an example of one of these studies that shows this. So what was done was a study in 42 species of Drosophila, in which they measured the temperature at which 80% of the males tested became sterile, which you can see in pink, and the temperature at which 80% of the males died, which you can see in black. And what you can see here is that for almost 50% of the species they tested, the temperature that leads to sterility is much lower than the temperature that kills the males. But of course, organisms are expected to respond to heat stress, and they can do so by moving to cooler places or remaining in the same place and either adapting to these new conditions or plastically responding to this new environment. And there have been several studies showing that these arthropods can plastically respond to heat stress, for instance, by changing male-male competition or increasing female mating rate, such that these females that mated with sterile males can somehow recover their reproductive loss. However, the majority of the species that studied these plastic responses are polyandrous. There are also some studies, although fewer studies, looking at the adaptive responses of these species to heat stress, and the majority of them show limited ability to adapt to these high temperatures. And here again, you have an example of a study done in 6 Drosophila species that were exposed to continuously increasing temperatures each generation, and you can see that eventually all of them become extinct. Again, the majority of the studies looking at the adaptive response of the species has been mostly testing polyandrous species. And this is unfortunate, since the mating system should be a key determinant of the impacts of temperature. Unlike polyandrous species, in monandrous species, a change in the mating rate imply a change in the mating system, which means there should be an extensive rearrangement of the selection pressures felt by both males and females, and which makes it harder to predict how temperature will affect these organisms. So we wanted to know if these monandrous species have the potential to respond to heat-induced sterility. And to do so, we used Drosophila subobscura, which is a Palearctic species with a wide range distribution and a model organism in evolutionary biology, particularly in thermal adaptation studies, due to the existence of a latitudinal decline of chromosomal inversions they have. Furthermore, and particularly important for the question we pose, they are monandrous, and like the majority of the species within the Drosophila genus. So we took advantage of the fact that 2 populations had been collected, one in the Netherlands and one in Portugal, and brought back to the Fly Lab back in Portugal. And they were kept in the lab for 70 generations at constant 18 degrees, so control conditions, optimal conditions for the species in the lab. And after these 70 generations, each of these populations was subsetted and either kept at the control conditions of 18 degrees constant or moved to a warming regime. And this was replicated 3 times. Okay, I have to be more assertive with the clicking. So here I'm showing you the thermal conditions they were exposed to. So the control temperature, the control treatment, as I said, remained at 18 degrees constantly, and the warming regimes were exposed to a daily fluctuation that increased every generation in mean temperature and thermal amplitude until it got to generation 20, at which point the individuals were exposed to a mean temperature of 13.4 degrees, as you can see, and a peaked maximal temperature of 29.4 degrees, at which point the conditions were kept constant for 25 more generations, so a total of 45 generations of warming. And we were lucky enough that when this was all ongoing, there was this master student, Afonso Grandela, that was particularly interested in this question. So he took this forward and he essentially tried to answer these two questions. Is there an effect of high temperature on male mating behavior and fertility? And if so, do females respond to this male sterility and rescue their reproductive success? And because of the conditions we had in the lab, we wanted to know if this was contingent on geographical origin and or thermal adaptation. So to test is the first question. He took the evolved populations that you can see there represented, and these males, the males from these populations, were either kept at their 18 degrees constantly, or they were exposed to heat stress by being kept at 31 degrees for 69 hours. And these conditions represent a sublethal heat stress because the mortality was not particularly affected, but we expected fertility to be so. And these males, after being exposed to stress, they were paired with a female, so 4 hours later they were paired with a female, and then they were paired again 3 days and 8 days later to see whether the males could recover from sterility. We measured several traits, and here I'm showing you two that are representative of the overall responses that we found. So on your left, you can see copulation latency, and you can see that males that were stressed take longer to start mating than males that were non-stressed. And this is true irrespective of the selection regime or the geographical origin. The same thing happens for reproductive success, where you can see that stressed males produce fewer offspring than non-stressed males, again, irrespective of selection and geographical origin. So this essentially tells us that males are, in fact, affected by heat stress. And then we wanted to know, as I told you, if they can recover from this stress. So you can see now appearing that the copulation latency decreased when they were given a second and third opportunity to mate, which suggests that there is some behavioral recovery. And again, this was not dependent on selection or biogeographical origin, which suggests that this change is plastic. However, they did not recover their reproductive success. You can see they continue to produce fewer offspring, even when giving a second or third mating opportunity. Again, no effect of selection or geographical origin. So now that we knew that males were indeed sterile and didn't particularly recover very well, we wanted to know if females could respond to this stress and somehow buffer this male effect. So what Afonso did was to take females that were not stressed and pair them with two males in sequence, the first male and then a second male 3 days later, And these males came from the selection regimes and again were either non-stressed or stressed exactly the same way as I described before. And as expected, when the females first mated with a non-stressed male, only 5% of them remated, which confirms the polyandrous mating system that they are supposed to have. But when they mate with stressed males, their mating rate increases up to 40% when the second male is non-stressed and 30% when the second male is stressed. And again, there was no effect of selection or geographical origin, which tells us that this shift in mating system is plastic. Okay, so now this is the last graph I'm going to show. It's a bit complicated, but bear with me. I think I'll be able to explain the whole thing. So then we wanted to know if there is indeed a benefit for females to remate. And what you can see here in the graph is a green dot and a black dot. The green dot represents all the females that did remate when given the opportunity to do so. So essentially they represent the females plotted in the first graph. And the black males represent the females that did not remate when given the opportunity to do so. And you can see here that right from the start, when females mate with non-stressed males, the ones that do remate are those that produced fewer offspring to begin with. And this pattern continues or remains when the females are exposed to stressed males first. And more importantly, when these females are placed with a second male, when they do choose to remate, the increase in reproductive success is higher than the females that choose not to remate when given that opportunity, which suggests that remating is indeed driving the rescue of female performance. So just some summary of what I just told you. We found that exposure to high temperature hindered the fertility of both male and female, the male directly and the female through the male. And there was no effect of selection nor of geographical origin, but males were able to plastically recover their behavioral performance, but not their fertility. And there was a plastic shift from monandry to polyandry, which should be crucial for population persistence under thermal stress. And with that, I want to thank you all and all the people that participate in the experiments, and I take questions. Thank you. Okay, so we did test a few temperatures below that, just to define what was this sublethal high temperature, because we wanted to test things, at the limit of, the limit temperature they would endure, let's say. But I can tell you that one thing that could explain the fact that there was no selection is the fact that the warming conditions they were exposed to during the selection process were quite different from the ones we applied when we were testing these responses, because they were exposed to daily fluctuations, and here they were exposed just to a brief heat stress that was particularly high compared to what they were experienced throughout, and this could explain this. Because actually, I don't know if I have time, wait, it's here, but it doesn't matter. We did test the reproductive performance of these populations under the conditions they were exposed to during the experimental evolution, and in those there is a response of selection. There is local adaptation, basically, in these cases.
Thank you.
You're welcome. Yes. I think it's both. We measured other things, as I told you. We measured latency to copulation, copulation duration. We also measured the frequency of droplet given, because this species provides a nuptial gift to the females. And I think both things are happening. I think males are a bit less good at courting the female, but the females are also less interested in mating. Thank you.
So, hello everyone. My name is Théo Bodineau. I'm a PhD candidate in Sorbonne University, and my PhD investigates plastic modification of breeding phenology in reptiles. And today I want to present you a result from an experiment that performed last year, which is about fitness consequences of breeding phenology plasticity with nocturnal warming and food restriction in lizards. So faced with climate changes, terrestrial ectotherms show important phenological changes, And a large proportion of these phenological changes can be due to the thermal plasticity of their reproductive cycle event. For many ectothermic taxonomic groups, we have limited knowledge about the consequences of this thermal plasticity of breeding phenology, and thus understanding the consequences of this phenological shift is essential to understand the vulnerability of ectothermic species to climate changes. So in a reptile, developmental thermal conditions have important consequences on life history traits of juveniles. And in general, warmer developmental thermal conditions have positive effect on juvenile by affecting the quality of embryonic development and consecutively the quality of juvenile produced, or by affecting the hatching date, so the birth date of juvenile. And in general, earlier birth date had positive effect on juvenile growth by giving them extended period of growth before winter, and because there are less competition, for example, in early summer. So despite the general pattern of positive effect of warmer developmental thermal condition on juvenile, little is known about the consequences of the thermal acceleration of reproduction in females. So in viviparous species, the viviparity is a critical life stage for energy balance for female, because viviparity induced additional reproductive costs such as an increased metabolic rate and increased energy expenditure due to embryonic development. Thus, the thermal acceleration of breeding phenology can increase energy expenditure over a shorter period of time, and thus could alter maternal homeostasis and reproductive output. Faced with diurnal warming, female can limit the potential negative effect of the thermal acceleration of breeding phenology by behavioral compensation, for example, increased food consumption. But little is known about the consequences of the nocturnal acceleration of breeding phenology, because during the night, behavioral compensation is very limited for diurnal species. And recent studies suggest that nocturnal warming could increase the physiological imbalance in these species. Additionally, while climate changes can have negative impact on primary production and prey availability, for example, for reptile, reduced prey availability during gestation could accentuate the negative effect of nocturnal warming. So in this context, we have investigated the effect of nocturnal warming and decrease in food intake on female and life history trace of juvenile in a viviparous lizard. So we predict that nocturnal warming should accelerate gestation, and that nocturnal warming and food restriction during gestation will have negative effect on reproduction and increased physiological imbalance in breeding female. We predict that nocturnal warming have, however, a positive effect on juvenile life history traits, and we predict that food restriction during gestation could accentuate the negative effect of nocturnal warming on female homeostasis and reproductive output. So we used the common lizard as study model, which is a small diurnal viviparous lizard widely distributed in northern Eurasia. And we both manipulated nocturnal temperatures and food availability in laboratory during reproduction in a balanced full factorial design. So we expose pregnant female to hot, moderate, or cold nighttime condition, and ad libitum food or moderate food restriction by giving them alpha food. And we then quantify the effect of our experimental treatments on maternal condition, reproductive output, and juvenile life history traits. So first, we follow the condition of breeding female during gestation, and we measure the lamp thickness, which is a reliable proxy of energetic condition of breeding female to assess reproductive costs. So we measure lamp thickness at the start of gestation and after parturition. And after parturition, we note the parturition date of breeding female, so the hatching date of juvenile. And we quantify reproductive investment as the total litter mass produced by the female. Then we measure the endurance of juvenile with a circular racetrack, and we release female and juvenile in semi-natural enclosures to follow growth and survival of juvenile. and we quantified the post-summer growth of juvenile using a recapturation. So first, we found interactive effect of nocturnal temperatures and food availability on female parturition date. So the nocturnal warming induced earlier parturition date, irrespective of food, except for female exposed to moderate night, where food-restricted female give birth earlier than well-fed female. So the high thermal plasticity of gestation is in line with several studies in reptile, but to our knowledge, no study have already demonstrated an effect of food availability alone or in interaction with temperatures on breeding phenology of reptiles. So food availability normally is unlikely to influence the speed of embryonic development, but this interaction can be explained by a subtle behavioral modification induced by food availability and temperatures, but this hypothesis requires further investigation. Then we quantified changes in lamb thickness during gestation. So here, negative changes means a loss of muscle, the muscle catabolisms. And we found an interaction of nocturnal temperatures and food availability on changes in lamb thickness during gestation. So under food ad libitum condition, we found that only female exposed to hot nighttime temperatures tend to lose lamb thickness, but under moderate food restriction, we found that female exposed to hot and moderate nighttime condition lost lamb thickness. So in viviparous reptile, food intake generally decreased during gestation due to the mechanical constraints of egg volume in the abdomen. Muscle catabolisms is common to recover amino acid requirements and energy requirements during gestation. And our results suggest that reduced prey availability could accentuate the metabolic costs of nocturnal warming of female condition. Then we quantify reproductive investment, and we found additive effect of nocturnal temperatures and food availability. So hot nighttime temperatures, as well as food restriction, reduce, on average, reproductive investment. So the common lizard use stored resources before winter and resources recently acquired during gestation or vitellogenesis to fuel reproduction. So a low food level and increased energy expenditure induced by hot night reduced the reproductive investment in this species. So then we quantified juvenile endurance at birth using a circular racetrack as the running time until exhaustion. And we only found an effect of nocturnal temperatures on this variable. So juvenile from female exposed to hot nights tend to have a higher endurance, than juvenile from female exposed to cold night. And our results suggest that hot nocturnal temperatures have positive effect on locomotor performance of juvenile and maybe the juvenile quality. Which is in line with several studies that show that warmer temperatures, developmental temperatures, positively affect embryonic development. And finally, we quantify juvenile growth, so juvenile body size after the summer, and we only found also an effect of nocturnal temperatures. So juvenile from female exposed to hot nighttime temperatures are larger than female exposed to moderate and cold night. But when we replace our thermal treatment by the birth date of juvenile, we found that the model is better adjusted, and we found negative correlation between post-summer juvenile body size and birth date of juvenile. So warmer nocturnal developmental temperatures have positive effect on juvenile growth by influencing their birth date, and earlier birth date have positive effect on growth by giving them an extended period of growth before winter, and probably because there are more favorable conditions in early summer with higher thermal condition and less competition between conspecifics. So to conclude, we found that nocturnal warming and food restriction had negative effect on reproduction and increased physiological imbalance in breeding female. We found also that the thermal acceleration of gestation induced by nocturnal warming has contrasting intra- and intergenerational effects, so positive effect on juvenile and negative effect on female. And we also showed that nocturnal temperatures and food availability can interact to drive breeding phenology, and that reduced prey availability during gestation can accentuate the negative effect of nocturnal warming. So overall, our results highlight the importance to consider nocturnal warming as well as benefits and costs of phenological shift in the context of climate changes. So I want to thank all my collaborators and the organizer of this symposium. Thank you for your attention. Yes.
I guess I was wondering, given some of the results about seeming benefits to juveniles of having heat-stressed mothers, is it almost like, but mothers produce fewer offspring, is that right? Fertility is reduced?
No, yes, they produce the same number of offspring, but the body mass of offspring is reduced.
I see. But the offspring have higher endurance?
Yes.
Okay, never mind, I got confused.
I guess I'll follow up because I was having a similar question. Do you have any sense of how those costs and benefits might weigh up, might balance in a longer term situation?
It's a good question. We haven't that. I think my hypothesis is that the nocturnal acceleration of breeding phenology can have positive effect in juvenile, in the early life, but maybe they can accelerate the pace of life of juvenile with more rapid growth. And then studies have shown that this accelerated pace of life have negative consequences for adult survival and reproduction. Oh, yes, short-term benefit, yeah.
If not, it's good timing to swap over the presentation.
Thank you.
Thank you so much.
All right. Hello, everyone. I'm Henry Rosato, and I'm going to be talking about how de novo mutation can help preserve species integrity in the case of the disruption of sexual signaling using digital evolution. But first, I would like to take a moment to thank my co-authors on this project, Dr. Mingzi Xu, an assistant professor at the University of Minnesota, and Swastik P. Pati, who's a PhD student. So now to begin. So as you probably all know, species isolation barriers are a critical part of maintaining species integrity. And one of the major mechanisms behind these species isolation barriers is sexually selected signals that are species-specific. However, anthropogenic changes may weaken species integrity by modifying sexual signals and/or signal preferences. And this is not just a hypothetical. There are, in fact, multiple studies examining taxons diverse as frogs and cichlid fish that show this happening in natural systems. There are many ways that these sexual traits can be modified by the environment. For instance, turbidity can limit female discrimination between males, as has been shown in cichlid fish, or temperature can change the mean values of sexual traits, as has been shown in the case of lions' manes. And, in addition, pollution can increase or decrease expression in ornaments and displays, and this has been shown in a wide array of taxa. And so the question I wanted to ask was, how can environmentally altered expression of sexual signals affect the maintenance of species boundaries over evolutionary timescales? And my null hypothesis was that species boundaries would be maintained via post-zygotic barriers without any need for any evolutionary changes. Alternatively, altered sexual signals could overwhelm post-zygotic barriers, leading to complete species breakdown. And finally, I consider the possibility that de novo mutation could allow for sexual signals to differentiate enough to preserve species integrity. Now, there are a number of challenges to actually answering this question in most systems. Number one, it is just very, very difficult to examine evolutionary dynamics real time with field studies. Obviously, there are many examples, such as with Galapagos finches, et cetera, but it is very difficult to find a system where you can examine really specific questions, such as this. And it can also be challenging to control for environmental variables when conducting experiments, either in the lab or in the field. And a final challenge is that even relatively long evolution experiments with animals that last over 50 generations may have difficulties in capturing the long-term evolutionary dynamics. So one potential solution to this is using digital evolution. In digital evolution, virtual organisms, bits of computer code, compete, mutate, and reproduce on a virtual computer, allowing us to examine evolution in real time. Digital evolution allows for fine-grained manipulation of the environment and very controlled experiments. It can last for thousands of generations, permitting the study of deep evolutionary history in a way that you simply cannot do with other systems under most circumstances. In addition, digital evolution has previously been successfully used to study sexual selection and species isolation barriers, although not in this particular context. Now, you might be thinking that digital evolution is very similar to simulation modeling. However, there are some key philosophical differences. One way to think about simulation modeling is that it is fundamentally a kind of artificial directed selection. You're trying to parameterize to get a particular result. And to show that, I'm showing these domesticated pigeons, where you're trying to select for specific traits and figure out how you can select them. By contrast, digital evolution is, in a sense, natural selection. It is fundamentally open-ended. You set the initial conditions, and then you just see what the system does. You don't know beforehand, in all likelihood. And to be clear, both are valuable, but they're different. And so instead of using a simulation approach, I want to use a digital evolution approach to explore the more open-ended aspects of this question. And so for our experimental system, I evolved 150 populations of digital organisms in the Evita platform for about 1,500 generations, or 50,000 updates. Each environment was seeded with 2 different species adapted to distinct niches in that digital environment. Niche differences form the basis of partial hybrid incompatibility, leading to post-zygotic isolation, but incomplete post-zygotic isolation. And finally, one species had an initial display and preference set to 1, the other -1. And these are arbitrary numbers. They don't correspond to anything real. They're just arbitrary values for these traits. And then we controlled several aspects of our environment. We controlled whether there was a stressor present. So in either, we would have the expression of the sexual signal would be increased for both species by the environment, or it would be decreased for both species. And the result of this would be the overlap between the signal of one species and the preference for the other, because the preference was not being modified in its expression by the environment, even though the signal was. We also modified the de novo mutation rate, so we had a control of 0, and then we also had rates of 0.0001 and 0.005 per copy. And finally, as a general control, we forced individuals in the control to mate only with conspecifics, preventing any hybridization. So we could see what would happen if hybridization was simply not allowed under these conditions. And so the results of this experiment, we estimated species differentiation over time using the k-means algorithm. And this is our overall results. So on the y-axis, you can see species differentiation, again, measured using k-means. And on the x-axis, time in updates. So 50,000 updates, about 1,500 generations. And what you can see so the lower half is the treatment, the upper half is the control without any hybridization. So I'm just focused on the bottom half where the species could hybridize. And you'll see that when stressors were introduced, mean display increased or decreased, initial species breakdown occurred. You see a dramatic loss of species differentiation. However, over time, at mutation rates greater than 0, species redifferentiated. Although if you see the gray mutation rate 0 line, you'll see that that never fully recovered. We also calculated the difference between sexual signal values between the species, basically how displaced these traits were. And so here you can see that overall they tend to increase over time, at least with positive mutation rates. Throughout all of them, they look like they increase a little bit more under the treatments, but overall they're generally increasing. However, at mutation rate 0, you'll see that when a stressor was introduced, any differences between the populations completely collapsed. So there was no trait differentiation over the course of evolutionary time between these populations because there was no de novo mutation. Finally, to look at this more closely and try to really tease out what was going on, we did Lixt linear effect model analysis. And so we looked at what was affecting species differentiation. And what we saw was that de novo mutation rate had the strongest effect on maintaining species integrity. So indicating that as the qualitative graphs that I showed before indicate that de novo mutation rate is really critical. And then here are the other things that we looked at, which are also all significant and are associated in ways that we would expect. We also looked at differences in signal between the populations, also using an LMM. And what we noticed is that the strongest effect was the difference in preferences between the populations. And that indicates that there was very strong sexual selection on this signal to match the preference of the females. And again, you can see here are the other things we looked at. Some are significant, some are not, but none of them have as strong an effect as the difference in preference. And finally, we decided to look at preference specifically, using, again, an LMM. And one interesting thing that we saw was that this presence of a stressor, this stressor that leads to that increases hybridization, was positively associated with mean difference. So that indicates that stressors were causing selection for increased signal divergence in these populations. And then you can see the others, and again, you can see the difference in display/signal is also positively correlated, very strongly as we would expect, and then everything else is non-significant. And so what is the big picture takeaway from this? To me, what this seems to indicate is that you have de novo mutation, and this can lead to divergence in signal preference, and then that leads to divergence in sexual signals. And what that does is permit rescue of species integrity even in the face of these stressors. And so, in summary, digital evolution is a valuable tool for examining the long-term evolutionary response of reproductive traits to ecological stressors. And in many ways, it can complement field studies and simulation modeling. Our results also show that de novo mutation can allow for evolutionary rescue of species integrity from these stressors. And populations' capacity for evolutionary changes in sexually selected traits should be considered when predicting the consequences of signal alteration on species boundaries. And I think this is one area where the results of this digital evolution work can be used to inform field studies of natural populations. And finally, I would like to acknowledge the members of my lab, the SHU Lab, for their feedback and support throughout their project. I'd also like to thank Dr. Michael Trevisano, who's sitting right over there in the audience, for clarifying the distinction between digital evolution and simulation modeling approaches to evolutionary biology. That was not an idea I had myself. I owe that completely to him. And finally, I would like to thank the University of Minnesota College of Biological Sciences Dean's Office for supporting this presentation through the CBS Dean's Office Travel Award. And with that, I'm happy to take any questions. Thank you. Yes?
Thank you. I'm wondering if you can talk a little bit about how recombination happens in the Evita platform and what you think and how we should think about recombination rate and how it might influence the evolution of the associations you see between preference and the traits.
Yes, totally. So you were asking about how recombination in Evita works and how that might the recombination rate might influence these evolutionary processes. So the way recombination works in Evita is that you basically have a, basically it's just swaps out parts of the genome every time they reproduce. So basically you have two gametes, and then they basically just swap parts of their genome. And so you have the same kinds of effects as linkage or whatever. It's not, Evita is fundamentally haploid, but it still has the same kind of recombination process that you might see in a regular chromosome. And as for how it affects the evolutionary trajectories, I would say that really is dependent on how much standing genetic variation there is. So if there's already standing genetic variation through de novo mutation, then I would assume that a higher recombination rate would speed up evolutionary rescue. However, I have not tested that, and that would actually be a really great thing that I would like to look into in the future. So thank you for that comment. Yes?
I'm wondering if, in addition to the mutation rate, if you looked at how the magnitude of mutation or the height of mutation affected your results?
Yeah, so, and that's not something that we looked into a ton, but what the results seem to indicate is that most of the important de novo mutation is directly related to the sexual signaling. So it's basically leading to increased divergence between the species. And I think that ended up being the mutations that really mattered, but I have not yet done a really thorough analysis of that. But that is definitely something I do want to look into in the future.
Thank you.
All right. Yeah, thank you all for coming. My name is Ivan, and I'll be talking about the effect of social environment and climate on the reproductive behavior of this little fish. I can't point at things, unfortunately. Along a latitudinal gradient, which is the coast of Norway. So this little fish is the two-spotted goby. It is a semi-pelagic fish, quite small, 4 or 5 centimeters in adult size. And you see a male and a female. The female is the one on top in the picture. Has an annual life cycle, and it's pretty common on the Atlantic coast of Europe and in the Baltic. It has male-only parental care, which is the most common form of parental care in fish. But the reason why I'm actually interested in it is because it's really undisturbed by human observation. So you can go in the field, and I don't know if the video is going to work. Yeah, and you can go in the field and you can look at courtship. So this is, so you see the female in courtship colors. She's transparent with the orange belly. The male is displaying his fins to the female, so that's a mutual courtship now. And you can really observe that from, so that was just in case the video didn't work, yeah. From very close quarters, so it's really cool for studying courtship in the wild. So I want to mention this study from about 20 years ago that kind of started sexual selection research in this species. That was done on the west coast of Norway by Forsgren and colleagues. So they went to look at the frequency of courtship events by males and females, and you can see on the little graph to the right. So to the left of that graph, you have courtship frequency by males that are represented with this little purple fin throughout the breeding season, which is between May and July. And you can see that the courtship frequency is decreasing a lot as time goes by. But the courtship frequency of females on the right side of the graph is increasing a lot. And so what that means is that, first of all, the courtship behavior is very flexible, or plastic, if you will, and also that this species is a great system for studying what is actually driving variation in courtship behavior. So what do we think is driving this variation in courtship behavior? A central concept here is the operational sex ratio that I represented here with a cartoon. So you have an adult population and you have a mating pool, which is basically individuals that are ready to breed. And you have now males and females, and you have adults that are also outside of the mating pool. So the idea is that the sex ratio in the mating pool, also called the operational sex ratio, is determining mating competition and sexual selection, in the sense that the sex that is the most common in the mating pool will be under more intense competition for mating, and therefore is likely to show more frequent courtship behaviors as well, and other behaviors as well. But today I'll focus on courtship. So the question is, if we observe fluctuations in courtship, does that mean that if operational sex ratio is driving courtship behavior, that there is also fluctuations in operational sex ratio, and what is driving those fluctuations? That's a pretty complex system taken all together. So what can drive operational sex ratio fluctuations? Of course, demographic parameters, so adult sex ratio. So you can notice that on this cartoon I represented more females than males, which is the case in most of our populations. This can occur, for example, if there is heavily sex-biased mortality. Another aspect is, of course, the mating system. So I mentioned that it's paternal care in that species, and the idea is that after mating, males and females have to perform different tasks before they're ready to mate again, right? And in this case, males have to look after the eggs until hatching, females have to produce a new batch of eggs, and those different tasks take different amount of time, which can create an imbalance in the mating pool. And of course, an overarching thing is that ecological factors can affect all the things in different ways. So for example, water temperature in fish can be very important for physiological pace and can affect the tasks of males and females differently, and also the operational sex ratio. So the idea was to ask the question, is the operational sex ratio driving courtship behavior? And then can we find effects of ecological factors on this whole system? So to do that, we went to the fields and we chose, so this is a map of Norway and a bit of the west coast of Sweden. We chose 6 populations, and each population, so we have about 10 degrees of latitude, which is almost half of the latitudinal range of the species. And we have 6 populations, and each is replicated 5 times. So we have in total 30 populations, and we went to each of those 3 times, early, mid, and late in the breeding season, to kind of draw temporal trajectories of the courtship behavior. Yeah, and so in practice, that means that we spent a lot of time squatting under the rain and snorkeling in very cold water, which was very fun. And then so every time we went to one of those populations, we looked at the courtship behavior, and we also looked at what I call here the social environment, but basically one aspect of it, the operational sex ratio. So first I'll describe the temporal trajectories of courtship that we observed in the different populations, and then I'll talk about the effect of operational sex ratio on the courtship and then ecological factors. Sorry, I went too fast. So here I show only the 2 southern populations to begin with. So the one to the right is the same as the one on the earlier study that I mentioned in the beginning. And what you have on the x-axis of those 2 plots is the time, so early, mid, and late season. And then on the y-axis, you have the courtship initiation frequency, either by females in yellow, so yellow triangles, and purple squares for males. So what you can see is basically a similar pattern to what they saw in the earlier study 20 years ago, which is early season, a lot of male courtship, late season, a lot of female courtship. So it's kind of cool to see that this is replicated across time, and that this happens in 2 populations that have similar latitude. Then if we go to the other populations, so I'll skip the middle one just for the sake of making it short. So if we look at the northern ones, there are about just above the polar circle, basically where the environment is quite different, and I'll come back to that later. You can see that for males, it's a similar story. It's a lot of courtship early in the season, which decreases over time. But for females, we didn't see a single event of female courtship. So it's 2 populations replicated 5 times, so 10 populations, 3 samplings, so 30 sampling events, not a single female courtship. So really something different is going on in those populations compared to the southern ones. The question now is, can we explain that with operational sex ratio? So to answer that question, we use a reaction norm framework where we basically say that the social environment or the operational sex ratio is the environment of the reaction norm on the x-axis. So in this case, more males to the right, more females to the left. And the trait that is plastically responding is the behavioral propensity. So in that case, I represented more male courtship when there's more males, and vice versa, which is what we would expect. And I say behavioral propensity, so it's not the same as behavior frequency, right? So what that means is that the propensity is when a male and a female encounter, what is the probability that a behavior will arise, right? So to calculate those propensities with field data, we have to correct for the encounter frequencies of males and females, which we do. So what do we find? Okay, this is a bit of a lot of stuff going on there, but I'll go slowly through it. So those are the reaction norms that we find. On the top, with the yellow trend lines, which are fit for a model, this is female reaction norms, and the bottom male reaction norms, and each panel is the time of the breeding season, so early, mid, and late breeding season. And so each of those panels is a reaction norm, meaning that the x-axis is a social environment, more females to the left, more males to the right. And the y-axis is the propensity to start courting. So you can see that we find some of our expectations confirmed in the sense that if you look at late season for females, so the top right square, you can see that the more females, the higher the courtship propensity of females. So that matches the prediction from OSR theory. And early season, so the bottom left, we also find that for males. The more males, the higher propensity for males to court. But what we did not expect to find is that the rest of the time it doesn't work. So early season, females don't do anything, even if there's a lot of females around. And late season, males don't do anything, even if there's a lot of males around. And there is data in late season males, it's behind the line and it's hard to see. So the question is, what do we do with that? Do I have time? Yeah. So first of all, it's a new perspective on an old result, because with this thing that I presented earlier, where they showed that male courtship frequency decreased and female courtship frequency increased, they assumed that there were constant reaction norms behind that and that the social environment was changing. But what we can say with what we have found today, which was only possible because we included many different populations, is that we actually have a change of social environment and a change of reaction norms, is sex-specific over time. Now, back to the initial question, which was, can we explain why there is no female courtship in the north? So to do that, we need to bring in ecological variables as well. So in this plot, you have two important variables for this system, which basically define the span of the breeding season. One is the water temperature, which is represented by the curve, the temperature in the breeding habitat for the different populations. So you can see that it's much warmer in the south, that's the red curves. And it stays warm for longer, meaning that the juveniles can grow for longer and be ready to overwinter. So the breeding season can go on for longer. The other one is the primary productivity season, which is basically when there is food available in the coastal ecosystem, and that's delimited by the two triangles. So when you're between the triangles, you can eat. Outside the triangles, you can't. And so you can see that in the north, it's also much more restricted, meaning that the breeding season is just overall shorter. What does that mean? It means that in the north, there is no late breeding season. And if you come back to the reaction norms that we drew and you remove the late breeding season, you see that there is no space for female courtship. So to explain why those populations show what I would call different dialects of courtship, you have to bring in those reaction norms, but also the environmental parameters. Yes, that's it. So in conclusion, does courtship behavior respond to operational sex ratio? I think yes, but not always the same, depending on the time of the breeding season. And if we want to speculate a little bit on why we see those sex-specific changes in reaction norms, so I have a couple of ideas. One of them is actually the individuals that we see in early and late season when we go out in the field are not the same individuals. So we could have early and late breeders that have different strategies and that also have different courtship propensities. And one reason to think that this is the case is because we have also placed artificial nests in the field, so we have data on males, and we know that early breeding males and late breeding males are phenotypically very different, so they're probably having different reproductive strategies. With this, I would like to thank everyone involved in this project, which is called Dynamar, founded by the Research Council of Norway, and in particular, I'd like to thank Sebastian, Lotta, and Trond, who is the head of the project, and also all the amazing people that made this huge field effort possible. Thank you for your attention, and if you want to find our work right now, it is on bioRxiv. Thanks. Yeah.
That's a really interesting talk. Thank you so much. I just had a question about the females courting the males later in the breeding season.
Yeah.
And I was wondering if you looked at maybe the quality of those females. So if they're different individuals and they're courting later in the season, are they maybe poorer quality females, and so they're needing to compete for the few males that are left to take care of their offspring?
Yeah, that's sorry. So that's definitely a possibility. On the top of my head, I cannot answer, but we do have a lot of phenotypic data, so I could check. What I know is that males are still breeding at that point, but there is not also a single time where a male has positively responded to a female courtship initiation in our observation, so it must be very rare. So we know that sometimes female courting must lead to a mating, but it must be quite rare.
Can you give us
I would like to do a common garden experiment, at least. So reciprocal transplant, you mean like we would have them in the field?
Yeah, to see whether the ones from the...
Yeah. So I think the best we can do is to be in the lab and do a common garden, because I don't, practically we couldn't have them in the field, like in a cage, I guess, or something. Or we could, but it would be challenging, yeah.
And would it be possible, I mean, to see, I don't know if it's related to that question, whether the females in the late breeding season have a better or worse reproductive success than the ones in the early breeding?
Yeah, I mean, it's very unlikely that late season is a good strategy, because they have to overwinter as juveniles, and basically the mortality is size dependent. So if you make offspring late, their survival is... The other aspect is that most of those guys are annuals, so they don't have a chance to breed a second time. So you could also wonder why they would stop breeding at all, because, yeah, is it time to... Yeah.